Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati, and in the demo video where the Banesh is going to discuss about the different aspect related to the chromatography system. In the demo what we have chosen the ActaPure M as a chromatography system, and we have discussed different aspects of the chromatography system in terms of the operation of the machine, and as well as how you are getting the different output into the computer and how you can collect the fractions. In addition, the Banesh has also discussed about the different precautions what you have to take while you are performing the chromatography utilizing the ActaPure M. And I hope you will enjoy the demo and you will understand the different aspects related to the chromatography system.
In this video, we will show you how to operate a FPLC instrument and the basic principle laying mechanism of how separation of the proteins using FPLC. FPLC nothing but fast protein liquid chromatography. We can say it is a derived version of HPLC. The main difference between HPLC and FPLC is FPLC can only be used for the separation of the proteins and sometimes small molecules also if we have columns available. But in HPLC we can use columns for separation of small molecules. Suppose if you have enantiomers, that also can be separated using HPLC. The columns what we will use for HPLC and FPLC also differs. In case of FPLC, we will use plastic columns, but in case of HPLC columns, we will use steel columns. Because the main reason why we are using like this is, if you use stainless steel columns in FPLC, because of the salts and high concentration of the salts and different materials we are using, it may corrode the steel. So that's why there may be improper separation of the component which we want to separate. So that's why we will use only plastic columns in this one. The separation of the proteins through FPLC is based on the size and shape of the protein. So if you have, like in gel filtration chromatography or size exclusion chromatography, in all those mechanisms like size exclusion, gel filtration can be applicable in FPLC also. So it depends on what column you are using. So if you want to purify histol proteins, you can use nickel NTA column, pre-packed column. Suppose if you want to only separate high to low molecular weight proteins, you can use any gel filtration column suitable for your protein. But the principle behind the separation is same. So these details of like size exclusion or gel filtration we are shown in previous video. In this video we will show how this instrument can be operated. In our lab we are using ActaPure. This is from GE Health Sciences. So all the component whatever we show, it is similar in other instrument or other companies instruments also, but only the architecture of the instrument changes. So let's see what are the parts it contains. So this is the instrument. It is connected to a system for observation purpose. So in this system it contains this stationary phase. This is the column. This is the mobile phase, that means buffers. This is the area where we will keep all the buffers. So it starts with the pumps actually. So these two are pumps, this one pump and two pump, these two sets. Whatever buffers are coming from this buffer tray, it will enter here. Okay, these pumps, whatever the pressure they are getting, they can be regulated here. This is the pressure monitor. This. So once the pressure is monitored, it will go to mixer. This is the mixer where two different buffers. Suppose if you are using if you are purifying through nickel NTA column, so in that case you need imidazole in a separate buffer prepared, and one is the equilibration buffer. In that case, if you want to elute that protein, particular histidine protein, you have to mix both the buffers A and B, for instance. So those buffers can be mixed here. Once the mixing is done, it will directly goes to the inlet loop. So once it entered into inlet loop, it will go to this chamber where it can be connected to the column. So the top portion of the column, it will connect here. We will show you how to connect the column in coming video. So after that, whatever it comes, it will enter here. And it will come directly into the UV chamber where the eluted component will be detected. So starting the instrument, there is a power button right side of the instrument. You have to just turn on that instrument. Then you can see a white light is blinking here. That means the system just started. So after that, we will go to this software part. For analysis of any elution, we can use this. It comes with the instrument. It is the Unicorn software we use for analysis purpose. So just double click on that one. And it will take you to the software. So it will give you three pop-up windows. One is method editor, another one is system control, and evaluation classic. So this is system editor where you can see chromatograms, and the other one is evaluation classic where you can analyze your chromatogram. Just go for system control. The first thing we have to do is connect the instrument. So here you can see the connected instrument Octapure 25. So just say OK. Now it will connect the system. So this will give different we can change different commands using this software. Just go to manual, execute manual. So this is the manual instruction software or dialog box where you can change things. So here different parameters you can change through this pop-up window, like pumps, flow path, and various parameters such as monitors. Just to go to pumps. So here you can change the system flow. So we can keep up to 20 ml if there is no column connected. So normal condition we can keep 5 ml also. So you just say insert this thing in order to execute it by system. Now here it is monitors, very important thing. We have 3 wavelengths here. We can monitor at 3 different wavelengths. So your choice you can give. So we are giving 280, 215 and 254. Just insert and say execute. So it started. You can see the green path is highlighted and also chromatograms appearing in the chromatogram area. So it will give 3 different chromatograms So one corresponding to blue, that is 280 nanometer for tryptophan tyrosine fluorescence. Second one is 254 nanometer for RNA or DNA related, and third one is 215 for peptide. So here we can see the path of the flow, how it is connected, starting from buffer A. So here buffer A, and it will go through the pump and mixing. Through mixer it will go to all the way to waste. So here different parameters we can change during running. We can change B also. If you want to change B, you just say start pump B. So see, we can see highlighted area. If you stop the program, it will save automatically. See, you can see some dialog box appears, preparing for new run. This is software introduction. These are the buffers we are going to use for this demo. The buffers need to be filtered through 0.2 micron filter and also degassed. For degassing purpose, we will use bath sonicator. So it will remove any air or air bubbles present in the buffers. It will remove those things. So we are using most common buffer, that is phosphate buffer with having pH 7.4, and this is the liquid water, and this is 20% ethanol. All the buffers were filtered through 0.2 micron filter paper and also degassed. So we have washed already, the system already being washed. So now what we will do, we will connect the column. So here precautions need to be taken while connecting this thing. So if you have any air bubbles through these loops or the piping system, it will directly enter into column, which will destroy it. So to prevent that, we have to make sure all the loops and pumps got washed thoroughly, and then we will connect in running condition. Before connecting the column, we have to remember few points. This column, whatever the bits are there, this is in 20% ethanol. So if you directly connect it, already ethanol is there. Whatever the flow rate we are giving, it will give more back pressure. So the bit between the distance between the column filter and the beads, settled beads, may increase. So that will reduce efficiency of the column. So what we will do, we have to change these pumps into water. So we changed it into water. Now we can connect it to the column. Here also some of the precautions need to be taken. If you are using chilled buffers, okay, suppose you need cold buffer, so that means you have to bring those all the components of the system to the temperature which you want to use for your purification. Otherwise, if you having a chilled buffer which directly enter into column, that may clog or precipitate some of the salts present in the buffer inside the column. So that will also reduce the efficiency. So this is also need to be taken care while running the FPLC. Before connecting the column, we need to adjust few parameters. So here is the software system flow. I am keeping 0.5 ml per minute since we are going to connect the column. So if the flow rate is more, then the pressure alarm may come. So after that we have to set the monitors. So this is also I am going to set. system flow 0.5 insert, and now we have to set alarms. At what pressure you need to get alarm. So I am keeping this 0.8 is fine. So once it is done, you can insert, then execute. So next here, this is the column connecting portion. So where here this is the upward portion where we have to connect with the column. Now we are not using column, so just we have to go to the column. We just click this one, column down flow. So from top to bottom, now you can see highlighted one. So you cannot directly connect to the column. First you have to fill the buffer or water in this loop so that there is no air bubbles. Just open the top of upside of the column. Okay, with this buffer itself you just directly connect. After connecting, you have to take out the lower portion of the column, otherwise it may burst also, but it is not the case, because if there is any high pressure, you will get alarm. So here this is the bottom portion of the alarm. You can see the buffer here if the buffer is passing through the column. So as we can see, there is a fill up of water in this thing. So once you see complete fill up of this loop or this knob, you can directly insert the lower portion. Once you see the buffer filling in, you just have to connect with the downward portion. So now the column is connected to the system and you need not to touch anything. Everything will be operated on the software. So here see, once the column is connected, you can see there is a change in the different UVs and conduction of the buffer. This is the red one is the this gray one is the for conduction, and that green one is the concentration of B, and these are three different UVs. Now we are washing with the water. So after once completely we removed 20% ethanol, then we will equilibrate in the buffer. So the main purpose of the equilibration is, suppose if you are prepared your protein solution in a, suppose say phosphate buffer, so you have water, you are not equilibrated with the phosphate buffer, then you cannot expect good resolution or good separation of the proteins, and also the proteins may not be stable in the other condition like in water, so they may degrade or they may not be useful if you are interested in the enzymatic reactions. So that's why we always do equilibrate the column with the same buffer which our protein of interest is dissolved. So this will also helps good resolution and keep the intact of the structure of the protein. So we are indirectly providing similar conditions for the protein, so it will behave in a native condition. So we completely washed out the ethanol whatever present inside the column. Now we will equilibrate in the phosphate buffer. So as we can see here, the conductance is completely comes to 0, and we can see the completely flat line, signal corresponding to UV280. So that means uh, there is no ethanol inside the column. In addition to that, we already washed 30 ml of water. So the total column volume inside the column is around 25 ml. So we also washed with 5 ml extra. So we can be sure that it is completely removed. Now what we have to do is we have to just pass the things whatever we set already without disturbing anything. So here we can see a pass symbol. You just pass, then come here and change the pitch to buffer. So once that is done, we will reset these things to continue mode. So we can see it is again activated. So we have equilibrated the column using equilibration buffer. So as we can see here, there is a stable line corresponding to UV280 and there is no other illusion coming out. So with this we can confirm that we equilibrated the column properly. So it's time to inject the protein solution and analyze it. So for injection purpose, this is the port where we are going to inject the solution, protein solution, and this is the loop. Whatever we inject through this injection valve, it will be stored inside this loop. The size of the loop depends on how much protein you want to inject and the column capacity. So we have 1300 HR column also. It is it can be used up to you can inject up to 1 ml. So in this column we cannot inject that much. If you want to inject, we have the company people might also give this kind of loops. So this we will connect as shown in here, and we will use for the injecting the protein solution. So what we will do is we have to set few parameters here. So here flow path, so injection valve you have to show it inject here, so insert this one. Okay, once that is over, we just have to insert the protein solution and execute the command. So as we can see here, the chromatogram here, the protein is eluted. So at 13 ml of retention volume. So if you want to identify the protein molecular weight or determine the unknown protein's molecular weight, you have to run this kind of analysis. Like you need to be known that what is the protein molecular weight, known protein's molecular weight, so which is available actually, commercially available. So you have to take that protein and just inject. Based on that, you have to construct a calibration curve between log molecular weight and the KAV, that is partition coefficient, which is calculated based on the elution volume subtracted with void volume divided by total volume subtracted with the void volume. That will give partition coefficient. So whatever you will get a graph straight line, and based on that straight line you can get unknown proteins molecular weight. So that can be discussed in size exclusion chromatography video. Now we will show you how to analyze the results. So this is the software used for evaluation purpose, evaluation classic. So you have to go to file, open chromatograms. So you have to locate where your file is kept. Just open that one and say OK. So here you are seeing so many things. This one corresponds to pressure and conductance. So you can customize the things like what you want to see in the chromatogram is only UV 280. So you just keep those things and remove all those things. Okay. And here also it is showing 380. You don't need 380. You want to take up to 65. So you just go and change the y-axis. So you can see this is the chromatogram. Peak is very sharp. So you can also integrate the peaks. So this is UV 280 integration. So just say so it will give the exact retention volume of the each and every peak, how many peaks are present. So here we can see 12.86. this is the major peak what we have. So with this you can analyze the results. So if you want to calibrate, you have to calculate retention volume for all the proteins whichever you are using for calibration and construct the calibration curve between partition coefficient and log molecular weight. So with this you can identify unknown proteins molecular weight. So in this particular FELC demo, we showed you how the instrument works, what are the different parts and what are the precautions we need to take while running the instrument, and how the software works and how to analyze these things. So another point we forgot actually, this is the fraction collector. So while your protein is emitting, suppose your protein is emitting at 12, so started at 12, you want to collect fractions from that time onwards till the end of the emission. So what you can do, you can use fraction collector also from main window. This is the fraction collector, it will automatically moves. So here it contains the sensor, but you need not to touch anything. Okay. So in main system control, here fraction collector, there is an option for fraction collector. How many fractionation you need to be done, when you need to stop fractionation, and how much feed to fractionation outlet valve. All these things you can set there. In addition to that, you can also set system gradient flow. Suppose you want to elute a protein with the gradient, you don't know at what particular concentration of imidazole if you are using nickel NTA column, or at what particular concentration the protein higher or lower molecular weight elutes. So with this you can just adjust the concentration and length. You have to give in suppose 60 minutes. So what system will do, it will over 60 minutes of time it will increase 0 to 50%. So you can do this one reverse gradient also. So first you will give 15 and time you can keep just suppose 1 minute. So from time when you start the system, it will starts with the 50% of the B and reduces to 0. So all these things will make you familiarize how the system works with the fast protein liquid chromatography. So hope these things will help you to achieve your goals in your research. Thanks for watching. Now we understand how a fast protein liquid chromatography works. So after using the instrument, we have to from buffer to water we have to change the valves, because if you are directly keeping in the 20% ethanol, it may the ethanol whichever present in the buffer and the proteins or salts present in the column, they may get precipitated and clog the column. So it is a better practice, first you change this buffer system to water, then wash thoroughly, whatever the salts present inside the column it got eluted, then again you change to the 20% ethanol for preserving purpose. That need to be remembered for better performance of the system.
I hope you have enjoyed looking at the demo and you have understood the different operational steps what is required to perform the chromatography. You can be able to modulate these steps, because it is possible that you may not have the Acta Pure M in your laboratory, you might have some other chromatography system. So, what I suggest is that you can actually go through with the manual, and the overall the steps are going to be the remain same, except that the positioning of the buttons and all other minute detail might be different. So, with this I would like to conclude our lecture here. Thank you.